Close your mouth. Pick any note. Any note will do. The pitch does not matter, and sustain it for 10 seconds. Hum it. Now feel where the vibration goes. It is not just in your throat. It climbs into your face, into the bones around your eyes, into the bridge of your nose. There is a buzzing in spaces inside your skull you forget exist. I want to tell you what is happening in those spaces, because what is happening is not what you think. You carry, behind your face, 4 pairs of air-filled chambers. Most people do not know they are there. You have lived with them your whole life and never thought about them. But they are there, hollowed out of the bone, lined with wet pink tissue, full of air that came in through your nose, and that will in another second be replaced by more air. The maxillary sinuses sit behind your cheekbones. Press your fingers under your eyes, on the soft tissue just below the bone. That is where they are, walnut-sized chambers, the largest of the 8. The frontal sinuses sit behind your forehead, just above your eyebrows. Tap your forehead with a fingertip. You are tapping the front wall of a hollow space. The ethmoid sinuses are buried between your eyes. Not single chambers, but a honeycomb of small cells, packed together behind the bridge of your nose. The sphenoid sinuses are the deepest pair, sitting almost in the dead center of your skull, behind your eyes, separated from your brain by a thin shell of bone. 8 chambers, 4 pairs, all hollow, all connected to the nasal cavity by small openings called ostia, all lined with mucosa, the same wet pink tissue that lines your nose. There is a long-running argument among comparative anatomists about why the sinuses exist at all. Some have proposed they reduce skull weight. Some have proposed they cushion the brain. Some have proposed they improve voice resonance. None of these proposals has settled the question. What is clear is that the chambers are highly conserved across mammals, and that they all share the same wet pink lining, producing the same gas. Whatever they evolved for, they did not evolve to be empty space. The biology inside them is too active for that. Most of what I was taught about these chambers, when I was a young man learning physiology, was wrong. They were treated as drainage passages, vestigial spaces, empty pockets between bone, of no special purpose, that mostly caused trouble, that swelled up during head colds, that filled with infection, that ached when the air pressure changed. I now think we had it backwards. They are not drainage passages waiting to get infected. They are not vestigial. They are resonance chambers. And when you hum, you are doing the thing they evolved for. Look, a resonance chamber is a cavity that prefers to vibrate at a particular frequency determined by its size and shape. Tap a wine glass and it rings at a certain pitch. Blow across the top of an empty bottle and it produces a particular note. The pitch is set by the geometry. Your sinuses are no different. Each chamber has a resonant frequency somewhere between 100 and 200 hertz, depending on the cavity and the individual. When you hum, you produce a vibration in your throat, pressure waves, oscillations of air, traveling at the speed of sound through tissue. Those waves travel up your pharynx, into your nasopharynx, and they enter the sinuses through the ostia. Once inside the chamber, the waves bounce around. They reinforce each other at the resonant frequency. The air inside the cavity, which had been still, becomes turbulent. It mixes vigorously. Now, the wet pink tissue lining the inside of those chambers is one of the most metabolically active in your body. It produces a gas continuously, day and night, awake and asleep, and the gas is called nitric oxide. 2 atoms, a nitrogen joined to an oxygen. A simple molecule, but one of the most important signaling agents your body possesses. The enzyme that makes it is called nitric oxide synthase. The form sitting in your sinus mucosa is the inducible kind. The substrate it uses is the amino acid arginine, yes, the same arginine you eat in protein. Your sinus mucosa takes arginine, runs it through nitric oxide synthase, and releases nitric oxide gas into the cavity, drop by drop, all the time. When the air in the cavity is still, the nitric oxide accumulates slowly and diffuses out at a low rate. When the air is turbulent, when you are humming, the gas is swept out of the chamber in a sudden burst. The next breath you take draws that burst of nitric oxide deep into your respiratory tract. Eddie Weitzberg and John Lundberg at the Karolinska Institute in Stockholm measured exactly how much. They published the result in the American Journal of Respiratory and Critical Care Medicine in 2002. They had subjects breathe out quietly through the nose. Then they had the same subjects breathe out while humming. The exhaled nitric oxide concentration was measured in real time using a chemiluminescence analyzer. The increase was 15-fold. Not 15%. Not 50%. A multiplication by 15. A single sustained hum on the exhale raised the nitric oxide concentration in the breath by a factor of 15. The effect was immediate. It happened within the first second of humming, and it dropped back to baseline within seconds of stopping. What does the gas do, mechanically, once it has been released? It diffuses across cell membranes. It is a small lipid-soluble molecule. It goes through tissue easily, and it binds to a target enzyme called soluble guanylate cyclase. That enzyme, when bound by nitric oxide, starts producing a second messenger called cyclic GMP. Cyclic GMP is the molecule that actually does the work in the cell. It activates protein kinases that relax smooth muscle. It dilates blood vessels. It signals across synapses. Nitric oxide is the messenger. Cyclic GMP is the message. Remember this. It will matter when we get to the pharmaceutical part. 3 Nobel laureates spent their careers learning to understand this gas. Robert Furchgott, Luis Ignarro, Farid Murad. They shared the Nobel Prize for Medicine in 1998 for figuring out that nitric oxide acts as a signaling molecule in the cardiovascular system. The journal Science named it Molecule of the Year in 1992. The work cracked open one of the most important communication systems in the body. And the gas they were studying, that gas, is the same gas your face produces and your hum releases. Now you might ask, does the pitch matter? The answer turns out to be yes, but mildly. The Karolinska group and others have looked at this. Lower pitches, somewhere in the range of 100 to 150 hertz, which is approximately the speaking voice of an adult man and the lower part of the speaking voice of an adult woman, produce more efficient sinus resonance and more vigorous nitric oxide release than very high or very low pitches. The exact optimum depends on the size of the individual sinuses, which vary from person to person. For practical purposes, you do not need to find your optimal pitch. Any sustained tone you can hold comfortably for tens of seconds will do the job. The difference between a perfectly tuned hum and a clumsy one is the difference between 15-fold release and, say, 10-fold release. Both are dramatic. The body is forgiving, and here is where the chain begins. Because the nitric oxide does not stop at your sinuses. It rides the next breath downward. You inhale through your nose. The air you draw in carries nitric oxide, 15 times the baseline concentration, fresh from the humming you just did. It descends through your trachea, branches into your 2 main bronchi, branches again into smaller bronchi, branches again and again until it reaches the bronchioles, and finally enters the alveoli, the tiny grape cluster sacs at the end of each airway where gas exchange happens. The alveoli are the size of a small grain of sand. You have around 500 million of them. If you spread them flat, the surface area would cover roughly half a tennis court. Around each alveolus is a mesh of capillaries, the smallest blood vessels in your body. Each capillary is exactly 1 red blood cell wide. The red cells pass through in single file, each one taking up oxygen and dropping off carbon dioxide as it goes. Nitric oxide does something specific in this part of the lung. It is a vasodilator. It relaxes the smooth muscle wrapped around small blood vessels, causing them to widen. When the pulmonary capillaries are exposed to nitric oxide, they dilate. The blood flow through them increases. More red cells reach more alveoli per second. The surface area available for oxygen uptake expands. There is a phenomenon in lung physiology called ventilation-perfusion matching. It refers to whether each region of the lung is receiving the right amount of blood for the amount of air it has. A region with lots of air but no blood is wasted ventilation. A region with lots of blood but no air is wasted perfusion. Healthy lungs continuously adjust the matching by dilating capillaries in well-ventilated regions and constricting them in poorly ventilated regions. Nitric oxide is one of the primary tools the body uses to do this matching. What humming does is dump a small extra dose of nitric oxide into well-ventilated alveoli, by definition the alveoli connected to the airway you just hummed through, and improve the matching for the next several breaths. You become, briefly, a more efficient breather. This is not theoretical. The same pathway is exploited by inhaled nitric oxide therapy in neonatal intensive care units. Babies in respiratory distress with collapsed pulmonary vasculature are given nitric oxide gas through a ventilator. The dose is much higher than what your sinuses produce, typically 20 parts per million, sustained in clinical use. The mechanism is identical. The dose is the only thing that differs. But the lungs are only the first system to feel the consequences. The vibration that drove the nitric oxide release is at the same moment doing something else. To a different organ, through a different channel. Run your finger along the side of your neck, just below the angle of your jaw. Beneath your fingertip, deep beside the carotid artery, runs the longest cranial nerve in your body, the vagus nerve. The wandering nerve, vagus in Latin, means wanderer because it does not stay in your head. It descends from the brainstem, exits your skull through an opening called the jugular foramen, runs down through your neck, branches through your thorax, sends fibers to your heart at every beat, threads through your lungs, descends to your stomach, your liver, your pancreas, your small intestine, your transverse colon. By the time it terminates somewhere in your gut, it has passed through nearly every organ that keeps you alive. The vagus is the main highway of your parasympathetic nervous system. The rest and digest branch, the opposite of fight or flight. About 80% of its fibers are afferent. They carry information from your organs upward to your brain. About 20% are efferent. They carry signals from brainstem to organs. Most of what the vagus is doing at any given moment is sensing. Now, the nerve does not float through your neck in some isolated tube. It runs through tissue. Specifically, it runs alongside the pharynx, near the larynx, just behind the soft palate. And those tissues are densely populated with mechanoreceptors, sensory endings, whose job is to detect deformation, stretch, pressure, vibration. When you hum, those tissues vibrate. The same pressure waves that rattle the air in your sinuses are agitating the soft tissue around your vocal tract. The mechanoreceptors fire, and one of the targets they signal to is the vagus nerve. There is a research professor at Indiana University named Stephen Porges who has spent decades studying this kind of mechanical vagal stimulation. Porges built a theoretical framework called polyvagal theory, in which a particular branch of the vagus is preferentially activated by social engagement signals, facial expression, vocal prosody, throat vibration. Some of his theoretical claims about evolution and emotion are debated. I do not want to overstate Porges, but the underlying anatomical observation he points to, that the vagus is mechanically stimulable from inside the throat, through the kind of vibration humming produces, is uncontroversial. A separate line of research, led by Kevin Tracey at the Feinstein Institutes for Medical Research, has demonstrated that vagal stimulation can modulate inflammation. Tracey identified what he calls the inflammatory reflex, a circuit through which afferent vagal signals trigger efferent vagal output that suppresses cytokine release in the spleen and elsewhere. The clinical implant version of vagal nerve stimulation, used for treatment-resistant epilepsy and depression, costs around $30,000 to put in surgically. The implant is a generator placed under the skin of the chest, with electrodes wrapped around the cervical vagus. What humming is doing, mechanically, is a low-grade, non-invasive cousin of that procedure. The route into the nerve is different, vibration through tissue, instead of electrical stimulation through wire. The effect is much milder, but the target is the same nerve, in roughly the same place. Once you start nudging that nerve, the consequences spread through every organ it touches. The most immediate effect, and the easiest to measure, is on your heart. The vagus has its hand on the brake of your heart. Specifically, the right vagus sends fibers to the sinoatrial node, a small patch of specialized cells in the upper right chamber of the heart that generates the electrical pulse for each heartbeat. The sinoatrial node has its own intrinsic rhythm. If you isolate it from all neural input in a petri dish, it will fire on its own at around 100 beats per minute. That is its baseline. Your resting heart rate is not 100 beats per minute. In a healthy adult, it is 60 to 80. The reason is that the vagus continuously slows down the sinoatrial node by releasing acetylcholine onto it. The vagus is the brake. As long as it is firing, your heart rate is held below the intrinsic rate of the sinoatrial node. When vagal activity rises, the brake squeezes harder. Heart rate drops a fraction of a beat, but the more interesting effect is not on the average rate. The more interesting effect is on the variation between consecutive beats. Your heart does not beat with metronomic regularity. The interval between successive beats is constantly fluctuating by milliseconds in a pattern that is partly random and partly correlated with your breathing. This fluctuation is called heart rate variability. Cardiologists measure it in milliseconds. Healthy hearts have high heart rate variability. Hearts that have lost vagal tone have low heart rate variability. The difference is not subtle. Low values are associated with cardiovascular disease, with anxiety disorders, with poor sleep quality, with major depressive disorder, and, in large epidemiological studies, with all-cause mortality. A heart that beats with metronomic regularity is, statistically, a sick heart. When you hum, vagal tone rises, and heart rate variability rises with it. This effect has been measured directly. A research group in India at the Government Yoga and Naturopathy Medical College in Chennai published a study in the International Journal of Yoga in 2010 in which subjects practiced Bhramari pranayama, the yogic breathing exercise that consists essentially of humming on the exhale, named for Bhramara, the bee, and showed acute changes in heart rate variability markers consistent with parasympathetic activation. The effect was measurable within minutes. It was not large, but it was real. The numerical changes are modest but consistent. Across the small studies that have measured this, the variability between heartbeats typically rises by 10 to 30% during and immediately after a session. That is not a transformative effect on its own. It is a real one. And it is happening because of the route I have just walked you through, not because of some abstract relaxation effect. Here is what is happening. All together. You hum. The vibration agitates your sinus mucosa, releasing a 15-fold burst of nitric oxide. The vibration also stimulates pharyngeal mechanoreceptors, which signal the vagus. The vagus fires more vigorously. The brake on your heart squeezes a bit harder. Your heart rate drops a beat. Your beat-to-beat variation widens. Your blood pressure ticks down a fraction. Your peripheral vessels dilate slightly. You are walking, by the simple act of humming for half a minute, from the sympathetic side of the dial toward the parasympathetic side. And the parasympathetic side of the dial is where sleep lives. I want to tell you something about how sleep actually begins, because most people misunderstand it. Sleep is not the absence of wakefulness. Sleep is the active replacement of one neural state with another. Different cells fire. Different circuits activate. Different chemical balances dominate. To fall asleep, your brain must transition out of a state in which a small cluster of cells in your brainstem, about the size of a grain of rice, on each side called the locus coeruleus, is firing the chemical noradrenaline throughout your cortex. As long as the locus coeruleus fires, the cortex is held in an alert state. You cannot sleep. Period. The locus coeruleus is part of what neuroscientists call the ascending arousal system. Other components include the nuclei of the reticular formation, the histamine-releasing neurons of the tuberomammillary nucleus, the orexin-releasing neurons of the hypothalamus, the cholinergic neurons of the basal forebrain. Together, these systems hold you awake. To fall asleep, every one of them must be turned down. Several things turn down the locus coeruleus. One of them is the activity of vagal afferents arriving at the brainstem nucleus called the nucleus of the solitary tract. The nucleus of the solitary tract sends inhibitory projections that modulate the locus coeruleus. When vagal afferents fire vigorously, the nucleus of the solitary tract quiets the locus coeruleus. The cortex de-arouses, drowsiness rises. Picture, for a moment, the person I am describing. The person over 50, maybe over 60, maybe over 70. The person who has been handed over the past few decades a stack of recommendations for sleep. Cut the caffeine. Limit the screens. Try meditation. Try breathing exercises. Try yoga. Try cognitive behavioral therapy for insomnia. Try a sleep tracker. Try cutting alcohol. Try a magnesium supplement. Try a melatonin gummy. Try a weighted blanket. Try a white noise machine. Try lavender oil. Try sleep hygiene. Some of these things help. Most of them are well-meaning. Some of them are frankly expensive nonsense. The person trying them all is not stupid. The person trying them all is desperate. Humming is not on any of those lists. It is not meditation. It is not a breathing exercise. It is not relaxation. It is not a supplement. It is not a device. It is not an app. It is not a habit. It is not something to be tracked. It is the mechanical agitation of air inside 8 bone wall chambers and the consequent release of a signaling gas that activates the parasympathetic branch of your autonomic nervous system through a specific anatomical route. It is just physiology, performed on yourself, in the dark, for 60 seconds before sleep. I want to slow down here, because I want to tell you exactly how confident to be in each of the 4 claims I have just made. There are 4 links in the chain. They are not equally well established. The first link, the 15-fold increase in nasal nitric oxide during humming, is the strongest. It has been replicated. The methodology is straightforward. Anyone with a chemiluminescence analyzer can reproduce it. Take this one to the bank. The second link, that the released nitric oxide reaches the lungs and improves pulmonary blood flow on the next breath, is well supported physiologically and demonstrated in clinical inhaled nitric oxide therapy. The exact magnitude of effect at humming-released doses in healthy adults is less precisely measured. The mechanism is on firm ground. The effect size for healthy people is modest. Trust the mechanism. Do not expect dramatic numbers from a single hum. The third link, that vibration in the pharynx mechanically stimulates the vagus nerve, is anatomically uncontroversial. The mechanoreceptors are there. They signal to the brainstem. The exact frequencies and amplitudes that produce maximal vagal output have not been rigorously characterized. We know the route exists. We do not yet know precisely how to optimize it. The fourth link, that humming, through this vagal pathway, produces clinically meaningful changes in heart rate variability and reductions in sleep latency, has supportive evidence in small studies of Bhramari pranayama. The studies are small. The methodologies are variable. The effect sizes appear real but are not yet established with the rigor of a large randomized trial. There is enough here to take the technique seriously. There is not yet enough to write prescriptions. Hold the 4 tiers in mind together. The molecular and anatomical claims are rock solid. The cascading consequences for sleep are mechanistically plausible and supported by small clinical signals. I will not pretend the top of the cascade is as nailed down as the bottom. But the bottom is unimpeachable. The biology of nasal nitric oxide release during humming is one of the cleanest results in respiratory physiology. Whatever else is uncertain, the gas is real, and the burst is real, and your face is making it right now. Let me show you something about pharmaceutical economics. The nitric oxide signaling pathway is one of the most lucrative drug targets in modern pharmacology. Sildenafil, the drug sold under the brand name Viagra, does not produce nitric oxide. Sildenafil works by inhibiting an enzyme called phosphodiesterase type 5. Phosphodiesterase type 5 is the enzyme that breaks down cyclic GMP. Cyclic GMP, as I mentioned earlier, is the molecule that nitric oxide uses to signal smooth muscle to relax. By blocking phosphodiesterase type 5, Sildenafil keeps the nitric oxide signal alive longer. The downstream signaling molecule is the same. The pathway is the same. Pfizer has earned, by some estimates, more than $30 billion on this single molecule. Tadalafil, sold as Cialis. Vardenafil, sold as Levitra. Avanafil, sold as Stendra. All of them work the same way. All of them amplify the same nitric oxide signal. Combined, this drug class generates several $1 billion in annual revenue. Vagal nerve stimulators are the next category. Surgical implants, small electrical generators placed under the skin of the chest, with electrodes wrapped around the cervical vagus nerve in the neck. They are approved by the Food and Drug Administration for treatment-resistant epilepsy and treatment-resistant depression. The implant procedure plus the device runs roughly $30,000 in the United States, depending on the system. There is also a non-invasive version that clips to the ear and stimulates the auricular branch of the vagus through the skin. These run a few hundred to a few $1,000s per device. The sleep medication market is the third category. Zolpidem, sold as Ambien. Ezopiclone, sold as Lunesta. Suvorexant, sold as Belsomra. The over-the-counter antihistamines repurposed for sleep, like diphenhydramine. The melatonin supplements. The magnesium glycinate sold for relaxation. The American sleep aid market alone, last I looked, was somewhere north of $70 billion annually. The global market is larger. I added up. The pharmaceutical and device industry generates over $100 billion per year, conservatively, on interventions that target nitric oxide signaling, vagal tone, and parasympathetic activation. These interventions exist for good reasons. They are stronger than humming. They are more reliable than humming. For people with severe insomnia, treatment-resistant depression, refractory epilepsy, or erectile dysfunction, the pharmaceutical interventions produce effects that humming cannot match. Let me be specific about what this does not mean. It does not mean humming will treat clinical depression. It does not mean humming will end an erectile dysfunction problem. It does not mean humming will replace a vagal nerve stimulator in a person with refractory epilepsy. The pharmaceutical interventions exist because the dose differential matters. A person whose nitric oxide signaling has collapsed cannot fix it with a hum. A person whose vagal tone is so impaired that they need an implant cannot fix it with a hum. The hum is a small, repeatable nudge for a system that, in most healthy adults, only needs a small, repeatable nudge to do what it is supposed to do. The pharmaceuticals are reserved for the cases where the nudge is not enough. Both are real medicine. They operate at different magnitudes. What I am telling you is that the molecular machinery being targeted by all of this expensive infrastructure is also being targeted, mildly, freely, side effect free, by 60 seconds of humming. Same nitric oxide, same vagus, same parasympathetic shift. The doses are mild. The pharmaceutical doses are dramatic. The pathway is the same. Your throat is doing, in miniature, what costs the rest of the world $100 billion per year. Here is something else I want you to consider. Humming is older than language. It almost certainly predates speech. Mothers hum to babies, have hummed to babies in every culture that has ever been studied, on every continent, in every time period for which we have records. Lullabies are, structurally, hummed melodies, soft, slow, low-pitched, in the range that maximizes sinus resonance. The mother does not know about acoustics. She knows that humming works on the baby. She has known that since before there were grandmothers to teach her. Religious traditions across the world have a hummed component. Gregorian chant, Hindu Om, Buddhist mantra, Sufi dhikr, Jewish nigunam, Tibetan throat singing, Native American ceremonial song. Cultures that share nothing else share this. It is universal in a way that demands explanation. People hum together when they want to feel calm together. They have been doing this for a long time. There is something else worth saying about why this works so reliably. Humming is one of the few things humans do that does not require a partner, equipment, instruction, or upright posture. You can hum lying down. You can hum in the dark. You can hum while resting your eyes. The very ergonomics of humming are perfect for the moment before sleep. This may be why mothers landed on it independently across thousands of separate cultures. It is the only sleep intervention that requires nothing but your throat. My mother used to tell me when I was a small boy and could not sleep, to hum a tune. Not sing, hum, with my mouth closed, until I felt sleepy. She was not a physiologist. She did not know what nitric oxide was. Nitric oxide was not understood as a signaling molecule until 1986. She did not know about the vagus. She did not know about the locus coeruleus. She knew that humming a child to sleep worked because she had seen it work on me and on my brothers and on every child she had ever rocked. What we are doing now with the science is not discovering the technique. The technique is older than science. What we are doing is decoding why it works. At the level of nerves, gases, second messengers, brainstem nuclei, the mothers were right. The traditions were right. We are finally catching up to them. So, close your mouth. Pick a note. Hum for 60 seconds. Not loud, not hard, soft and steady. The way you would hum a tune to yourself in a quiet kitchen at night. Let the vibration find your sinuses. Let the gas release. Let the wandering nerve receive the mechanical signal. Let your heart rate drop a beat. Let the locus coeruleus quiet. Let the parasympathetic side of the dial come up. You are not doing nothing. You are running, in the privacy of your own throat, an intervention whose molecular pathways are shared with several billion-dollar drug classes. You are doing what mothers have done with babies since there were mothers and babies. The hum is the oldest medicine. It is sitting, free, inside the bones of your face. I am curious. When you hummed, where did the vibration land first?